to go next, and they crawled under, and as they rolled out, a tiger snake popped up, a little neonate, and so I grabbed it, and I remember taking it back, and Adam was pleading with his dad. It's like, please, Dad, can I keep this wild tiger snake? It's like, okay, son, you're 15, you're old enough now to have a tiger snake. So Adam got to take that home. Do you remember that?
I do. I'm surprised you're admitting that.
Yeah. Well, also, the first snake I ever bought was Adam, who was clever enough to breed his diamond python with his coastal carpet python at the time when no one was able to breed anything. And I had to part with $40 to get 2 babies. You did. 20 bucks for a snake was a lot of money back then. Yeah. So you gave them away. To everyone other than me who paid $20 each for it. So it's great to have Adam back after so long and his long and distinguished career, and we'll be hearing all about the work he's been doing with skinks and in particular the genetics with the Agonias. We are still finishing, so the next Red Belly Courier is the 10th edition. We thought we'd have it out for May, then June, then July. Now we're hoping it'll be out by October. The majority of the book this year is going to be featuring Jerry's history of the AHS, which is going to have the first 3 decades. Next year we've got the AHS's 75th anniversary, so we've been trying to think about what we can do to commemorate that. Rick's got a new book coming out. You've got So Many Snakes, So Little Time. I was disappointed in that book, not for what Rick did, because Rick did an amazing job, but the production quality was pretty average, and it deserves so much more than what they delivered. So we've put Rick in touch with Reed New Holland. They're picking up his next book, which will be out within the next few months, called Snakes Without Borders, and we'll be launching that later this year, another night at the Riot Eastwood. We thought though that 2 events, one after another, just featuring Rick and his new book might be a bit too much. So we're going to try and incorporate it into Rick's book launch, the 10th edition of the Red Belly Courier. Chris has kindly agreed as of half an hour ago to come back and talk about the Northern Territory book. There was a lot of people who couldn't make it here, so we'll be repeating all that at Riot Eastwood and treating it as a fundraiser to get the cash to produce a really nice large book for the 75th anniversary of the AHS. So that's all in the process of happening. Jerry and I have been chatting about it. If anyone who was around in the old days, Adam, got any photos of anything from back then, Glenn? We just need a whole bunch of images to make this book everything it should be. Chris was around back in the early days too, so Jerry's the man to talk to if you think you might have anything that could contribute to that. But it'd be fantastic to see a really nice big publication, depicting a pretty amazing 75 years. Kane's not here tonight for the Red Belly Courier. I did speak to him a little bit earlier. We're hoping to have it finalised in the next couple of weeks and then going to print so we can make sure we've got it at this event come October. Field trips, anything planned? Frank?
Not yet. Just been discussing something with the people in the back row.
Mm-hmm.
So thinking about Smith's Lake. I will be overseas for quite some time, halfway December until the end of January, in Europe, visiting family, but perhaps try to organize a Smith's Lake trip before that time, end of November or so. I've had a meeting with the SOPA Rangers also, the Sydney Olympic Park Rangers, but we haven't finalized any dates for the surveys there yet. But the expectation is that there will be one at the end of September and another one at the end of October.
Okay, very good. So for those of you who don't know, this is a paid gig for the AHS. We've got an arrangement with SOPA, the Sydney Olympic Park Authority, where every year we'll go out twice and just survey whatever's happening with reptiles. We keep saying to them, if we can go and throw a bit of tin out the back, to what's going on than if we just walk around aimlessly, but
That
they don't
is-
want kin. That's another story. Okay. What's that?
Asbestos.
Yeah, asbestos is even better.
It's most likely going to happen because they've been doing an external survey over there, and that's one of the recommendations. So not the asbestos, but at least something.
Yeah.
Or pit traps or something.
Just throw some tin around.
Yeah.
Like there's so much of that area that is off
Yeah.
display or away from the public. If they just started throwing some tin around, they'd have a much better idea of what's there. So the AHS email's been down for the past week. It's now back up. I made the mistake of putting a 1-star review on the people that did our current website, and we've had problems since then. Anywho, so the new website should be up and running within about a week, and we've now got the emails back up as well. So if anyone has sent an email in the past 2 weeks, we weren't ignoring you, we just weren't getting it. Have you guys been receiving the email that Liz is now sending out, alerting you to these evenings? Put a show of hands, who's getting the email? Okay. Alright, 70-ish%. Have a chat to Liz. Liz is about to raise her pencil there, so you can all see who Liz is. We'll make sure you're on the list and we'll try and figure out what's going on. But yeah, hopefully we'll have that all sorted very soon. Lastly, Anthony's got Wild Expo 2.0 happening soon, and he'd love to tell you about it now.
Oh.
Yeah, he didn't know he was telling you.
Yep, we're doing a Wild Expo Reptile Festival on the 8th of October at Fairfield Showground. Should be fun. Looking for volunteers too, if anyone wants to come and volunteer. That'd be good. And just to make it all happen, we've got limited time to bump in and limited time to bump out. And it should be fun. I'm digging up Robo-Croc and a few of my cool toys. And yeah, just try and make reptile expos as educational as possible. That's the aim. And yeah, that's the main. They'll have all the elements of some of the Herpetological Society expos, but with the Wild Expo twist to it. Yeah, so it's just a 1-day event. This time, normally my expos are like 3 days, and they broke the bank. So hopefully we're going to not break the bank this time. Yeah. But yeah, Ryan, looking for helpers to want to help and volunteer. That'd be really cool. Thanks. Yeah,
Cool.
thanks, Chris.
Okay. Alrighty. We'll hand over to Adam now. You can take over. Please put your hands together. Cool.
Lights off?
Yeah.
Yeah, lights off, thanks. Well, yeah, thanks very much, Chris, for the invite. When you were talking about the early days, got me reflecting, got me thinking about it. And you know what, this society, I think, has had a pretty large impact on my career. It really is a wonderful society, and I think it's contributed a lot to a lot of people over many years. And you know, I'm sort of thinking about that, and I'm thinking, well, you know, I joined the society, and I learnt a lot about reptiles, being a passionate reptile lover. I ended up getting some jobs as reptile keepers, thanks to Anthony out at the African Lion Park back in the day when it was still up and running. And that then led to other jobs out in other wildlife parks. Strangely enough, Glen Shay's still sitting up the back like he was in the 1980s, and I think to a small extent, well actually probably a large extent, he inspired a scientific sort of curiosity in myself that then led to a career in science. So I'm going to talk a little bit about some of the work that I've been doing. Most of the work that I do these days isn't actually herp-based. So I'm a geneticist, a conservation geneticist, and I apply that to a whole bunch of different animals. But in terms of herps, I've done a fair bit of work in arid Australia on a few different species. I've done a fair bit of work on the Agurnia group, and over the last sort of 10 or 15 years, actually close to 15 years, I've been spending a fair bit of time in the Amazon basin looking at poison arrow frogs and some of the snakes over there. I'm at Macquarie Uni, as you can see, and I lead the conservation genetics lab. We've got a group of somewhere between 8 to 10 PhD students at any one time working on a whole bunch of different things. So let's see if I can run this. Right. So I'm going to start off talking, and I'm not going to talk about phylogenies. I'm going to start off just talking about the Gurnia group. There's a couple of different phylogenies out there for them. This one's from Gardner 2008. There's a more recent one. There's a few minor issues with them, but the only reason I've got this up here is, and I would reckon most of you probably know, that they're a really interesting group because you've got lizards that have different graduations, if you like, of sociality. And so, initially, as I'm sure you're well aware, Taliqua rugosa, the sugarback, was found to be monogamous. The late Mike Bull sort of pioneered that work and pointed out long-term monogamy in the sugarback. And then, following that, some people started using genetic techniques to look at some of these funny Gurnia groups that live in rock outcrops. And so there was the Stokesii, Gidgee skink, and Cunningham skink. And there was Mike Gardner and was looking at Stokesii, and myself, and I was looking at Cunningham skink. And I was looking at Cunningham skink. I'm sure you all know what Cunningham skink looks like. It's sort of distributed across much of southeast Australia. It's probably a species complex. I was looking at them on the central tablelands of New South Wales, and my primary goal was to actually look at what the effect of land clearing was, to see whether if you take away the vegetation, whether you end up getting a change in the genetic characteristics. So this was sort of coming from a conservation genetics bent, and it's following the idea that if you have small, isolated populations, then you're going to lose your genetic variation. And if you lose your genetic variation, well, that's the stuff that you need to evolve, okay? So you start losing your adaptive potential. You start losing your ability to adapt to environmental changes. So that's one of the problems. The other problem that I'll elaborate a little bit further is that you can then get inbreeding and inbreeding depression. So I'll talk about that in a moment. Anyway, I ran around and I caught hundreds and hundreds of Cunningham skinks, mostly in Elliott traps, and I did some genetic work on them. And what I found was that when you take the vegetation away, that they tend to disperse less, and you tend to get more highly related individuals in close proximity in these rock outcrops. So they seem to be spending more time sort of stranded on these rock outcrops surrounded by grass. So that was the finding, and then of course, conservation genetics theory, you get a whole bunch of related individuals together, well, they must be inbreeding, okay? And if they're inbreeding, okay, if you've got a whole bunch of related individuals that are breeding with one another, then genetic variation should go down, and that's bad. That didn't happen. So I did the measurements. Oh, okay, there's no difference. Okay, this was part of my PhD. This was in the late 90s, early 2000. Okay, no difference. Well, that's a shame. And it was then that I was sort of looking at the arrangement of kin groups in these rock outcrops. That this was kind of a side issue for me. I was more interested in the conservation genetics bit, but I delved into the behaviour and found that there was long-term monogamy and long-term site fidelity, and they live in these family groups. Okay, so when we're talking about long-term monogamy, we're talking about across many, many years. And in fact, some of these sites, so I've got a site that I I started working at the late 90s. I've been back and had a look. It was probably about 2015. Most of these animals are microchipped. There's the same pair poking out of the same rock crevice after all that time. So they really do show very long term. The other thing, this has been found, I mean, this sort of pattern, has occurred across a couple of the different Agurnia groups. So it was being unveiled in Agurnia stokesii pretty much the same time. But another thing that I discovered is that if you actually look at the relatedness between pairs of Cunningham skink, monogamous, they're in their rock crevice, been there for years, you have a look at the relatedness between them and other potential mates in the area, and it's substantially lower. So somehow these individuals are actively choosing mates that are lower than you would expect, just kind of randomly choosing another lizard out there in close proximity. So they've got active inbreeding avoidance, and that was like, oh, okay. Well, maybe on the time scale that I've been looking at habitat fragmentation, maybe on this time scale, this inbreeding avoidance behaviour has been sufficient to offset any evidence of inbreeding, right? So that was a finding that I found quite interesting, other than just the behaviour. At the time, it had just been, a similar sort of pattern had just been found in Agurnia stokesii, but it was fairly new. This sort of behaviour was a fairly unexpected sort of behaviour in a reptile. Okay, so they're avoiding inbreeding, and I guess also if you just sort of think about the investment that these individuals are making, as I said, you look at a rock outcrop and you'll see, a monogamous pair of lizards that have been producing offspring regularly for decades. And they're a long-lived animal as well. So, so they're investing in offspring year after year after year. So if they're putting so much investment into their partner, then it kind of makes sense that they want to choose a partner that's going to maximise the health, the fitness, the health of their offspring. To me, that makes sense. That they may have evolved a inbreeding avoidance strategy given those sort of behavioural traits that they've got, that long-term monogamy. Sort of ongoing question, whether the inbreeding avoidance they're carrying out, whether it's based on some sort of genetic cue, some sort of, I haven't done it with Cunningham skinks, but the Bull Lab did some wonderful work with a whole bunch of these things, and they looked at tongue flick rates, and they got Agurnia stokesii to flick their tongues at related lizards and unrelated lizards, and they could see that they could discriminate on the basis of smell. So they can use their smell to discriminate between related and unrelated individuals, but whether that's based on familiarity, because they live in family groups, or whether it's based on a smell that's based on some sort of genetic cue, that's still a question that we would like to get at. So this inbreeding avoidance stuff, I just want to sort of, in a fairly, I don't want to bore the living daylights out of here, but just in a fairly straightforward way, just talk about what some of the issues are. So in any population that you look at, except for a few very rare examples, there's going to be deleterious mutations out there. I'm sure I've got a whole bunch, and I'm sure most people carry mutations in their DNA, in their genome, that are deleterious. Okay? They're not great to have if they're expressed. Now, if you've got an organism that's diploid, i.e. you've got one set of chromosomes that come from mum and one set of chromosomes that come from dad, then you've got potential to have two different alleles at a particular locus. Okay. Sometimes, in many cases, the allele, if it's coding for a trait, could be recessive, which means that it's not expressed if the other allele is present. Alright, so most of the deleterious alleles that you have are recessive. Okay, in other words, you won't have the problem, you won't have the issue that's related to that bad allele, that deleterious allele, if you've only got one copy. However, if you get two copies, okay, you get the recessive allele and it's deleterious on chromosome from mum, and the recessive allele and it's deleterious from a chromosome from dad, and you're homozygous, then you'll have that deleterious trait, and that could affect your ability to survive and reproduce. And that's what inbreeding depression is. So as the genetic variation in a population goes down, and the level of homozygosity, that is having the same allele on both the maternal and the maternal chromosome, goes up, then you've got a better chance of actually having two bad recessive alleles, and better chance of that affecting your fitness, your ability to survive and reproduce as an individual, and sort of translates through a weaker population. So that's what inbreeding depression is. So all populations, more or less, have these bad alleles, these deleterious alleles, and they reduce the fitness of individuals and the population when genetic variation goes down. Okay, albinism, for example, is a classic sort of recessive trait, just by way of illustration. Okay, so you've got inbreeding also, the other point that I'll just quickly make, you've got inbreeding that happens as a result of genetic variation declining in the population, and also through matings between related individuals. Okay. So that's the issue of inbreeding depression. Okay, the next piece that I want to talk about is the Great Desert Skink, or Jacara, or Warrener, depending on which part of Australia you're from. If you're not familiar with them, they're a fairly large skink, you know, sort of a little bit larger than a Cunningham skink. They vary a bit in colour, so they can be brick red, they can be brick red with a whitish belly, brick red with a yellow belly. You get these grey forms, and you get a couple of different varieties on that. They're quite spectacular lizards. They really are quite spectacular. They're also vulnerable. So this is sort of roughly their distribution. What this is actually showing is the various sites that I've gone to and collected them from. And in fact, this site here in Wattaroo is in the northern APY lands in South Australia, and it was thought to be locally extinct. And when we heard that it had been spotted, fortunate enough to go out and actually get samples from this population in the northern APY lands. So I travelled around collecting great desert skink. You know, as I mentioned, they're threatened throughout much of their distribution, highly fragmented distribution. And I'll talk about some of the threatening processes in a moment. But you know, not only are they culturally really important for a lot of the First Nations peoples in that part of Australia, they really are. They absolutely cherish them. So not only are they culturally significant, but they also create amazing habitat for a whole range of other species. The mulgara, which in itself is a threatened species, actually often makes use of the burrow systems of great desert skink. So do womas, for that matter, and a whole bunch of other herps do as well. This is just an image of a burrow opening with you can see the Jacara footprints going into the burrow system.
Sorry about that, folks.
You're right.
Gotta find them
Right, so I've had a couple of different students work on Great Desert Ski. So I've had Siobhan Dennison, she did a PhD with me on Great Desert Skiing out at New Haven, which is an Australian Wildlife Conservancy property in the Tanami Desert. And Steve McAlpine, he actually wrote up the action plan for Great Desert Ski. He's been working on Great Desert Ski for decades and decades. In fact, in APY lands, he's known as Wadi Jakara, the Jakara man. And he's got a wealth of knowledge on Great Desert Skink, and he's got the eye to find. They're actually not very easy creatures to find out in the field. So they've got this burrow system, and so the burrow system tends to have multiple openings, interconnected burrows. They're deep, they go down deeper than a metre, so 1.2 metres. And like a lot of the Agurnia group, they have a latrine. So if you keep your eyes open, you might spot the latrine, which is usually right in the middle of this complex of different burrow openings. The burrow openings might span 10, 12 metres across. So they're very, very extensive burrow systems. They're often also associated with these termite pavements, and sometimes like fairy circles. And so when you're looking across a landscape, you'll see this sort of denuded circle, and that's where you've got a termite nest, subterranean, and favourite tucker for Jakara. They eat the termites. So you often see the burrow systems actually quite closely associated with the termites. Just as an aside, I'm collaborating with someone from CSIRO that's actually using the scats as a means of extracting DNA. So rather than catching the lizards, which I had to do, Steve and Siobhan and Danae, who I worked with as well, who also did a PhD with me in New Haven, talking in Elliott traps mostly. I'll show you a picture in a moment. But it's actually having quite a bit of success genotyping individual lizards from the DNA. Some success. It's not an easy task. From DNA that's present on the outside of the scat. Of course, it degrades fairly quickly under the ultraviolet light of the harsh conditions of arid Australia. Yeah, so you can see the Elliott traps here sort of spread out. That gives you an idea of the extent of the burrow system. So they really are quite extensive. Would bait them, and of course you then have to control the ants so that the ants wouldn't get into the Elliott traps. It kind of looks ridiculous cramming so many Elliott traps around, but it worked, and they're not particularly easy creatures to catch. This was actually in Wateroo, so this was the recently discovered population that was in an area where it was thought to be locally extinct. So with Michael Carney from Melbourne University, we collaborated with Danae from Australian Wildlife Conservancy to have a look at their optimal temperatures and have a look at the burrow system. As I said, the burrow system, is really extensive and it's deep. This is at New Haven, and the surface temperatures, they get high, 70 degrees sometimes. So obviously lethal surface temperatures during the warm times of the year. Right, so there was a paper that came out about 2011, 12. It's a good paper, I'm not knocking the paper, but they did actually hold up Great Desert Skink and say, hey, climate change is going to be an issue for great desert skink. Michael Carney gave me a call and said, "Yeah, I'm not so sure. Can we have a look at this?" I said, "I'm not so sure either. Those burrow systems are pretty impressive." And so anyway, we went and put data loggers in the burrow system and measured the temperature. And as you can see, we've got quite a range of temperatures here. So it's really hot on the surface, but it does a really good job of buffering the temperature there. It also does a really good job of maintaining the humidity, which is also obviously very important in that sort of environment. Okay, so get some extra sort of models and predictions, and came to the conclusion that actually, in this particular case, and this isn't a general rule, but in this particular case, climate change is not in a clear-cut way, certainly on the basis of temperature anyway. There might be other changes that are problematic, but in terms of temperature, it's not going to be the issue for great desert skink. If there are issues, it's threatened. Right into that. So that's the burrow system. So went around and you saw the ridiculous numbers of Elliott traps that are whacked around all the burrows, and you caught lots of great desert skinks, jacaré from these burrow systems. Then we looked at the relatedness of individuals within those burrow systems, and it's kind of like the Cunningham skink. You've essentially got family groups living in these burrow systems. The only slight difference is that the males tend to sire offspring in a few different burrow systems out to maybe about 500 metres from, do actually travel a fair way. Yeah, highly related. They're not a social lizard, and they tend to cooperate to actually maintain the burrow systems, which is quite interesting behaviour for a reptile as well. So you get multiple individuals actually in there excavating out the burrows, cleaning the burrows, and essentially cooperating in this rather large investment. You can burrow system like that, that's a lot of energy that they're putting in to maintain those burrow systems. Anyway, we don't need to worry about this one so much. This is just making the point that the males do in fact wander around and sire offspring in different burrows out to about 500 metres apart. So threatening processes, the obvious ones. That creature is certainly a threatening process. Fire regime is very, very important for great desert skink as well, and in fact both predation and the interaction between fire and predation was the main topic of Danae Moore's PhD thesis at the Australian Wildlife Conservancy. Another problem are hard-moved beasts, so cattle and camels. They walk onto the burrow systems and they just collapse them, and that's another threatening process. So they're key threatening processes. As I mentioned, Danae, should have a picture of her, but she did some amazing work out at the Australian Wildlife Conservancy. So she's actually one of the managers out there. The Australian Wildlife Conservancy New Haven site has been in the news a bit lately. It's got this massive exclosure where they've released bilbies recently. But it's actually one of the strongholds for warriner, as they know, in that part of Australia as well. There's a really large population there. And Danae and Danae's partner Joe more or less started managing New Haven after it was bought from BirdLife Australia, and they built it up right to this point of getting the exposure there. Danae had 2 kids and managed to complete her PhD while at New Haven, which is about 300 k's from Alice Springs in the middle of the Tanami Desert. So quite a remarkable woman. And Anyway, as I said, her thesis was primarily on threatening processes, and she carried out these very time-consuming experiments where she carried out controlled burns of different intensity, and she had tracking rings, and she looked at the presence of predators that were coming in post-fire or in unburnt habitat. Anyway, nuts and bolts of it are that burning isn't particularly great for the grey desert skink, and you know, after clean burns in particular, you can see that a whole bunch of the burrows that were previously occupied become unoccupied. I think most likely they're gone, they're dead, because that's a lot of investment to put into a burrow system like that. So fire regime is very, very important for these guys, and there was an interaction as well. You know, as soon as you burn, maybe this is the key factor, as soon as you burn, the predators come in, and particularly the cats come in and knock them off. Rightio, so with Juckera and Warriner at the moment, as I said, you've got these fragmented populations. The populations are dwindling very quickly. So in 2012, I was working with Steve McAlpine on the ball fields, which is a sand plain just near Uluru. And there really were quite a few Juckera there. I think had well over, I know it was well over 50 burrows mapped out just in the ball field region near Uluru. Pretty much gone now. So they are still disappearing. The populations are still dwindling on our watch. So one of the strategies that they're talking about is translocations. Now we know when we've looked at the genetics, as you saw on the map that I had at the beginning, you know, I've sampled them from all around the place. And I know that when the populations are small, the genetic variation is low, and that makes them prone for inbreeding depression, and inbreeding depression will accelerate, and it's been shown across lots of taxa, will accelerate the pathway to extinction. Right? So there is that issue of inbreeding depression, and we've got these populations that are dwindling very, very quickly. Something needs to be done. The land managers, plenty of the land managers from various groups are interested in the idea of translocating lizards from larger populations into smaller populations to supplement the individuals and bolster the genetic variation to offset that risk of inbreeding depression, right? And that brings me to the next thing that I'd like to talk about, and that is the potential risk of outbreeding depression. Like if things are too different. You don't want to produce a mule. So as I mentioned, I've sampled these. I've got samples well collected. When I say we, you know, the people that I was working with have samples from across a fair way of distribution, most of the latitudinal range. And what we know so far from the genetic work that we've done is that they're genetically distinct. Basically, these different colours mean that they're genetically distinct at those different locations, right? So you've got genetically distinct groups, probably means the level of migration between those groups, the level of gene flow between those groups is fairly low. And the next question is whether they're locally adapted. I mean, that most probably are. I mean, you've got different environments, so it seems that they most likely are. But what we'd like to do is actually measure that, get a good handle on it, so that we can then evaluate the risk of outbreeding depression, which we'll talk about in a moment. And then that can guide which are the best source populations to use for translocation if we want to sort of supplement the numbers of individuals and the genetic variation in these dwindling populations. to read, but the main point here that I want to make with outbreeding depression. So outbreeding depression, if you've got, groups that are locally adapted and you bring them together, then you disrupt these sort of locally adapted gene complexes, and the individuals that result aren't as well suited to the environment and they get selected out. And you see it all the time when you've got like hybrid zones and they're fairly tight, and you've got selection that sort of keeps these fairly tight hybrid zones. That's the consequence of outbreeding depression in the natural system. But it's not quite the issue that people sometimes think. I mean, it can be an issue, particularly with frogs actually, it can be quite an issue. But it's probably been overstated to a fairly large extent. I'll come back to that. This here is a decision tree that's been put together to help conservation managers evaluate the risk of outbreeding depression. And essentially, if you don't have any good evidence, strong evidence of local adaptation, and if the populations have been separated by less than 500 years on that scale of generations, then you're very unlikely to have outbreeding depression. Now this is just an excuse kind of to show some pictures of frogs from the Amazon basin. This is Alavates femoralis. It's one of the poison frogs distributed across a lot of the Amazon basin, and this is a very complex environment with large rivers that split up the distributions, and actually quite a variety of different habitats that also can act as filters or barriers to dune flow across the Amazon basin. Anyway, if you go through, a lot of these Alavates femoralis, these little poison frogs, they call out during the day, they're diurnal. A lot of them have been separated from one another for a long period of time because of the rivers and so forth. And genetically, they're different enough that if they were to breed, then there'd be the problem of outbreeding depression. And so they come up with little signals to tell each other, hey, if you breed with me, the offspring's not going to be any good, so just avoid it. And they involve little colour patches, okay, so yellow or red, and if you do trials, yeah, they will assortatively mate. They'll only mate with the same colour, right? So the other thing that they use, and if you're familiar with Little John's work, this was first behind this whole sort of idea of reproductive character displacement and calls was developed here in Australia. But anyway, you've got the same sort of thing happening here, where you've got these different vocal repertoires. You have 3 little beeps or 2 little beeps, and the calls are also an advertisement to say, hey, okay, I only breed with a 2 beeper, or I only breed with a 3 beeper. So they assortatively mate according to how many times they beep and also on the basis of their colour. Okay, and that's to avoid outbreeding depression. Right? And if you look at the hybrid zones where these different Alavates come together, they're very, very tight. We're talking about a big geographic area, and you only get hybrids across just a few kilometres where they come in contact. And the other one, this is a spectacular frog, right? Most of the poison frogs are small. You kind of think they're going to be big, but they're tiny little things, right? This sometimes they call a splashback frog. Babies like to notice, and they're quite big. They're about this big. Ridiculously colourful. These are all the different colour morphs that you've got across the Amazon basin, and I've been fortunate enough to go to most of these places and collect them. Bright orange, bright blue, and some of these colour morphs are actually very, very close to one another. You can walk through a bit of rainforest and you come to what is a relatively small river for the Amazon basin. You cross it, you'll go from bright yellow to bright blue. And it's a real enigma because these are poisonous. We've done the test, you look at the skin is toxic. The bright colours there is an advertisement, they're aromatic, it's an advertisement saying don't eat me. For an advertisement to be effective, then it should be the same. So a predator learns, but it's not. So there's quite an enigma here, and I've spent quite a few years trying to unravel what's going on, and that's another story. But what I can tell you is that they do assortatively mate on the basis of colour. Again, it's probably a means of. So the different colours, even though they're in relatively close proximity, are genetically very divergent. They're all one species. But they're quite genetically divergent, even though they're in close proximity, and it's most likely a way of avoiding outbreeding. Rightio, so I just wanted to sort of touch on the captive side of things just for a moment, and a lot of what I'm going to say is just conjecture at the moment, I love talking to groups like this. I know because I was part of this group in the 80s that there's a wealth of knowledge out here. So I just want to know what you guys think about this. But I can tell you what science says about a few aspects of captive management. One is that there is rather rapid adaptation to captivity, and in some species it's been shown that you can get adaptation to captivity in as few as just a couple of generations, right? So this is a thing that happens. So much of us, have reptiles at home that will be breeding for generations. Chances are there's been selection happening, and that they're adapted to captivity. So that's just a point that I'll make. So there's likely to be some sort of adaptation to captivity in the animals that you've had for generations on end. The other thing that often happens is there can be a little bit of inbreeding if you're not consciously managing, with the stud book or whatever, the individuals that you're using to breed with. And I mentioned what inbreeding is. This is when you've got these deleterious alleles that are unmasked when you've got low genetic variation, and it's been demonstrated in different reptiles. So some really wonky sperm, and there's very poor fertilisation success in these inbred pine snakes from Florida, and you get a whole bunch of other abnormalities associated with inbreeding. One of the first wild studies of this was by Madsen on Swedish adders, and there's a whole bunch of malformed young in these inbred population, I should say, and then he introduced males from another genetically diverse population. All of a sudden, everything was fine, and they weren't inbred, and the population went up, and it was genetically rescued. In a captive situation, I think inbreeding could be a real issue. And I've been out of the game for long enough now that I'm not really sure where people are at. But certainly back in the 80s, it maybe wasn't considered a great deal. The issue of inbreeding rather widely. Maybe it's considered more like it now, but I'd say that is quite a risk in a captive situation. So you've got adaptation to captivity, potential risk of inbreeding. Seems to be a few slides missing, but that's all right. I can continue on. The point that I wanted to make was with respect to outbreeding. Okay, so there's a lot of interest in the herb world and coming up with different colours, hybridising and getting these interesting looking reptiles. The other thing that's come to my attention is that, I mentioned it before, there seems to be a lot of emphasis on the problem of outbreeding depression, and it's been around for some time. So in the 90s, there was a lot of concern, about provenance. Like if you're collecting seeds for bush regen, you had to worry about provenance. And it crept into a few of the different organisations. It's like, oh, okay, now, if people keep these animals and they escape and get into the wild, that's going to cause some genetic pollution and it's going to cause a problem with outbreeding depression. And that's a little bit of a mantra that I've heard quite often in various government departments and authorities. And I think it's overstated. It's just conjecture, but I think it's overstated because if you've got a whole bunch of animals that are adapted to captivity, they're going to get selected out of the environment pretty quickly if they escape and get out in the bush. Now, if you guys are the experts, if you guys reckon that's not the case, I want to hear it. But my guess would be that animals that have been bred in captivity for a long time don't really pose much of a risk if they're getting out of the bush. Likewise, if you've got an animal that is a hybrid or isn't from the region and it gets out, there's a risk that it could cause a problem, but I think the risk's pretty low. And again, I gaze around the room wondering if anyone knows of any good examples of where, is there anywhere around Sydney where you see more hybrid carpet diamonds than diamonds? I don't know, I don't see them.
He does. He picks
No,
them up
Yes, sir,
every
do you?
One spot.
Okay.
Yeah, Loftus.
So you see hybrids.
dozens of
In
them.
the last few years, probably got 32, which are pure carpets.
Really?
And I've rocked up twice now to carpets breeding with mating with diamonds. At the time, I've turned up to the call out. Yeah.
Okay, well you're making my point. That's why I like to talk to people like you, because I get information from people that know.
Yeah. It's only that place in Sydney I found it up.
You ever seen carpets in breeding balls too? There'll be
Yeah.
a bunch of diamonds and there'll be a
Yeah.
carpet in there
Yeah, with
beating
carpet
the
making,
shit
yeah.
out of the diamonds.
Yeah.
So I would also wonder, if that was the case,
I just want to know who brought all the carpets in the area.
if that's the case, and maybe that's a one-off example. I don't think it's happening commonly.
No, it's not, no.
But if that is the case, then it's a problem. We've
Yeah.
got large-scale human changes to the environment.
The only thing I did find is that when it came to the breeding season,
Yeah.
the diamonds don't combat a little bit. The carpets, they combat a lot.
That doesn't
And they're
surprise me.
smashing the shit out of the diamonds.
That's interesting.
Yeah.
Yeah. Still, that's really interesting.
Yeah.
But still, I'm not convinced
But
that.
I'm getting from all sizes hatchlings too. All sizes, different colour relatives.
That's good. I'm glad you told me that.
Yeah.
But I'm still not convinced it's a massive problem.
Oh, no, me neither.
Yeah. And so when it's used as an argument that you shouldn't be keeping animals that aren't in the local region because there's that risk. To my way of thinking, I think the risk is fairly low from a genetic perspective, and it's often held up as a reason why one shouldn't.
Just look at the success that we have with releasing captive populations in the wild for conservation. Almost never works. Even
Yeah.
when you release hordes of them, adaptation to
going
captivity
nowhere.
happens so quickly that they're too susceptible in wild.
Well, you're the expert on that. Yeah, look forward to your talk. But yes, exactly. Anyway.
I usually find there's a reason those animals aren't in that area because there's a problem with the area. And when people keep releasing animals back to the area, there's obviously a reason they weren't there to start with.
Yeah, well, that's true. I mean, there is a with invasive species. There is a thing with chronic milk pressure. If you get enough in there, it can sometimes take off. But anyway, I think the risk is probably overstated.
I mean, if you think about buggery gulls, well, the commonest birds kept in captivity, is there any cactus budgerigars genes got into the wild population.
Yeah, that's a good example too.
I mean, we're talking about vast numbers of wild budgerigars, vast numbers of captive budgerigars. There would have been 1,000,001 opportunities.
Are we now getting blue budgies out west? Yeah.
Okay, so I guess I could sort of formally finish the talk just by saying I wanted to just highlight, the risks of potential risks of inbreeding, but I also really wanted to make that point about the overstatement of the risks of outbreeding, particularly when it comes to entrenched ideas about what might happen when organisms escape from collections. And I'll leave the formal bit of the talk there, but if you've got any questions, I'm happy to attempt to answer them.
Put some lights on.
Yeah, whack some lights on.
So then you can.
More.
You would think that the risk would be lower of escaped pets when you've got a similar thing that they can hybridise or inbreed with than if there's nothing filling that niche. So if you've got something that is totally unlike your local fauna, for instance corn snakes,
Yes.
getting out in Sydney, they can fill a niche that isn't actually there. But the chances of things sort of, introgressing into the population, I agree, are pretty low. I think that it's a phony argument, really.
I think I probably agree with the mention you mentioned corn snakes out there. You hear these rumours all the time about wild populations of corn snakes. Like people like Rob and myself and everything, we're professional snake catchers in Sydney, and the only corn snakes ever turning up are in apartment blocks and things like that. You get one or two, and I've never ever seen with my own eyes wild populations of breeding corn snakes. It's only.
I don't know if there is. I was just.
I don't, no, but I'm just saying that it's what you said there. It's what you hear all the time.
Yeah.
The people that have just got into the hobby, and they go, oh, there's wild populations in back of Sydney or Western Sydney, and they're all these. I said, prove it. I've never seen them. Like,
We just looked at 300,000 wires records from the whole of New South Wales, and there's no corns.
No.
A lot of very weird things.
And I've got corns, but like I say, every corn's been one individual animal. I've never seen anything that's
Speaking
proved
to
that
what
they're
you're
breeding.
saying, I initially just sort of threw the corn snake as an example of something that
I've had
might
probably
find a niche.
half a
But
dozen
what
bowls.
they're saying is that there is no niche for a corn snake probably.
Yeah.
But say there was, the corn snake would take off.
Well, that's all
But
the big,
the corn snake probably doesn't have a niche. Even though there's big hype about corn snakes and DPI would
Yeah.
love to make a giant hype about corn snakes.
Exactly.
As you say, unless there's a niche available to it,
Yeah.
But something
they're up against some serious competition for that niche.
Oh, absolutely.
And also they've got some serious,
Black snakes that love eating corns.
yeah, and they've got some serious issues to deal with these kookaburras that can find a worm under the ground.
Oh, yeah.
And
It's our corn snake friends.
yeah, I think one of these kookaburras around corn snakes is but something like maybe green iguanas might find a niche. They could unleash, but
Just like water dragons, you see them over the flood.
but speaking to what Adam was saying about the carpet pythons, you found what's basically happened to the diamond pythons in that area at Loftus is they're changing colour.
Yes. Yeah,
Temporarily,
and they're
mind
coming-
you,
Well, the funny thing is,
potentially.
I'm not getting hardly anything turning up looking like a hybrid. I'm getting pure coastal still turning up, and I've only seen adult pure diamonds mating with pure coastals. But I've never seen anything that's shown a hybridisation between the two yet. In actual in anything turn up, I've still only seen pure coastal babies. And yeah, so I think it's been very selective as to what's producing it.
But the other
Very
thing about the other thing about that, say that population of carpets with less and less appearance of diamond in them, this is where climate change can kick in too, because obviously diamonds are a temperate climate, species, everything about them reeks of trying to be as warm as possible,
That's
flat.
Yeah.
And as it gets warmer and warmer, then the carpet
Carpet
is going to have a
species.
selected advantage
Yeah,
over the
living
diamonds.
area. And yet,
Yeah.
So, and when you're
carpets-
saying that they're selectively.
No, there are some species.
What do you actually
Yeah.
mean?
But why weren't they
Essentially,
here
if
earlier?
they were
Why did that not take
wild
that?
Like if you bring 10
Up
colours in,
time.
break those five.
I'll leave it one. Yeah, Bernard.
Well, they were doing that for retics, so when I first started working in Toronto back in the early 90s, retics were just renowned for being super aggressive, and in America the retics were always aggressive. And then it was when Bob Clark started breeding them, anything that hatched and was bitey got bumped, and within 20 years all the retics were puppy dogs. They were so problematic for the first 30 years, and within 3 generations you can do anything with it at no time at all.
Likewise.
I was going to say the diamond snakes, west of the ranges from Kempsey High to the Macleay River. I've seen a diamond snake there, 100% pure. One of the prettiest diamonds I've seen. And living in that area too, there's no mixture. There's the most beautiful carpet snakes there too, from North Queensland, not from North Queensland, from the Queensland border.
Interesting, the same area.
In the same area, within a mile of each other.
That's really interesting. Yeah, okay.
They're beautiful snakes, the diamonds. Yeah. You were saying genetics before on the river in the States there, the Amazon. All those frogs are different colours, and they're genetically slightly different.
They're quite different.
Okay, genetically different,
Yeah.
but you're calling them the same species.
Yeah.
So what's the,
Okay.
how big, how much genetically different?
Thank you very much. No, that's a really good question. You've asked a question. So, the level of genetic divergence is substantial, but not of a degree that you would expect between. Okay, so if you look at the mitochondrial DNA, for example, the mitochondrial DNA is only a few base pairs different. It's actually not that different. It's well within the sorts of levels of divergence that you would expect to find within a single species. If you look at mitochondrial DNA and look at parts of the mitochondrial DNA that are kind of like barcodes, they're invariant. There's no difference. They all, the sequence is exactly the same across all of those frogs. So that's cytochrome oxidase, cytochrome 1. So same. So, but when I'm talking about genetic divergence, I'm talking about parts of the genome that evolve pretty quickly. And so we're talking, okay, so I timed it. So we're talking about the divergence, patterns of divergence that happen during the Pleistocene. Okay, so it's probably not enough time for speciation, but it's certainly enough time for there to be enough differences that have accrued that there's fitness consequences for the offspring if they reproduce.
So do you
Yeah,
think
but
you're
you're-
observing speciation? Well, if they're not interacting, they're on the way to becoming.
Yes,
Yeah.
and I mean the Amazon basin is known to pump out quite a few species. And I think that part of it's that habitat heterogeneity and the rivers and that complex history is all part of it. But the question you asked was exactly. I mean, that initially I was just like, "There's no way
Well, I'll give you
these
an
are a
example.
single species."
Paul Horner did his PhD successfully with very hard reviewers, like Alan Greer. He was spot on with his work, in my opinion. And Paul separated two lizards which are identical in colour, identically in different, but they're living in a totally different environment, and was accepted as a species with his PhD. And genetics meant nothing. And the same thing goes with genetics. I believe in genetics to a certain degree. I'll give you a typical example where I disagree, is with two turtles. I don't know if you're familiar with turtles or not, but Australis, they're still arguing about what is Australis in the Regent River area, where I believe it came from. And Victoria, which is 1,000 kilometres away, they're so morphologically different, distinct. No one can, but George's is the same. Their genetics are pretty close. They're the same species.
Yeah,
That is ridiculous, you know. There's no connection with each other in any way. So it makes you think with genetics, you know.
It's a complex and evolving field. There's a lot of variables happening. I hear what you're saying.
Yeah, I was having fun. So this was really interesting. You did a really good job explaining. The inbreeding, it's interesting what you're saying, because I was going to put forth, so the two examples are probably the most inbred reptiles that then became the most invasive reptiles.
Yes.
The beak, Burmese pythons and red-eared slides.
Yep.
Yeah. So it's just interesting that has that inbreeding and adaptation to captivity, has that actually some traits that allow them to actually become that invasive?
I mean, there's a whole journal on
Yeah.
invasive biology that talks about this kind of stuff. But there is, you know, a sort of a survivorship fallacy as well. You know, most don't thrive with low genetic variation, but a few manage to. Just because there are a few examples doesn't necessarily mean that it's a common thing. One of the things, okay, and then you can dig into it a little bit deeper. So what can happen when populations get small? If they're small for long enough and don't go extinct, then a lot of those deleterious alleles can get selected out. It's called purging. And if the conditions are right, all these deleterious things can get purged so that there isn't much left. And in that context, inbreeding depression may not be such an issue. Cheetahs, for example, are that genetically similar. You can take a skin graft from a cheetah in one part of its distribution and whack it on a cheetah in another part of the distribution. But they went through a historical bottleneck, and it was a long-winded one, and they purged a lot of the deleterious alleles and enabled them to persist. So there is some nuance there. So there's some subtlety there, and purging is part of the story.
Yeah.
And then there's a survivorship fallacy as well. And then there's epigenetics, which is another area that people are starting to look at a lot. The mechanisms that change gene expression, also the distribution and how that might impact invasive species as well. So there's quite a lot of detail there. But as a general rule, inbreeding, bad.
How many generation? Because I'm thinking about rough-scale pythons, and essentially we're talking, what, 5 animals or something. Is it, how many generations does it normally, would you expect until you're starting to see the house of cards potentially tumble, or is it?
It's a very active area of research at the moment, and there's not a lot of empirical data. It's mostly based on simulations. But the reason it's kind of hard to come up with there's a rule of thumb I can give you, but it's hard to come up with an exact answer, because it's an interplay between the length of the genetic bottleneck, the length of the population, length of time in generations that the population's been small, how small the population is, and the history, the deleterious bad alleles that population's actually carrying. And there's an interplay between those factors.
So you could argue that potentially that roughies do already have it in the wild, very small.
Maybe they're purged, I don't know. I mean, you'd have to, maybe, it's a very good observation, maybe because they are such an isolated and small population, maybe they have actually just historically
Similar thing
purged
you said for
themselves of deleterious alleles. It's quite possible, yeah.
In Australia, probably the one of the most inbred are corals.
Okay.
That's black snakes are from a very small group. When I used to keep collets and used to breed, that was the theory, because some of them had these like double ventrals. This weird stuff. Because the collets are all supposed to come from three.
Pose is describing that as a new species.
Sorry, it might be a silly question, but is it guaranteed that an animal has to have deleterious alleles? Can it not? Could you not just have two perfect animals?
Yeah, it's a very good question. Most individuals carry. I mean, the DNA that any one individual's got, everything that it needs to make itself as an individual. There are millions and millions of bases. They're the bases that make up DNA. It's incredibly complex, and there's a lot of it. So the chance of it being perfect across that whole genome, all that DNA, is very, very slight. Most individuals carry deleterious alleles. But as I mentioned, because most of them are recessive, if you've got a normal allele on that other chromosome, you'll never know. I mean, I'm sure I've got heaps, but they're masked. They're just not expressed because they're recessive. So, yeah, they're very tough. You think of every time in your germline, every time a cell divides, it's got to do it perfectly. Well, it doesn't. Mutations arise. So there are different levels, but it's highly likely that there's bad stuff in there.
Yeah.
Anything else?
I just can I get your assessment of this assumption, or have you got some assessment?
No, you go and then.
You go.
Actually, thank you. It was just a question about the fact that already the desert skips were showing, despite the fact that it was kind of like the continuous ecological zone almost, that they were already showing quite a bit of separation, or is it not like a continuous ecological zone?
Subtly different.
Okay.
And so they tend to sort of, most of the paleo drainage areas, and it's got a particular soil composition. It's usually this sort of plains, smithex plains in these old paleo channels, and you've got to have the association with the termites as well. So it looks, you've got to get your eye in.
Yeah.
It's actually quite subtle, but there are these sort of subtle environmental differences that greatly affect the suitability of habitat for Jahara. So I think they are naturally fragmented.
So they're patchy even ordinarily.
They're patchy ordinarily, and that's partly to do with the soil and the soil and the biotic factors. And I think historically, well, for periods of time, fire regime as well, throughout much of their habitat, we've also created suitable and unsuitable bits of habitat. So all of those things interacting, I think it's safe to say that, and the cultural information tells us also that they've been fragmented for a long period of time. Yep.
That's it.
No, I was going to ask Adam his assessment of this idea, and that is.
Put me on the spot, aren't you?
No, it's just an assessment of an idea. You can tell me that it's silly or not. So, for example, we were talking about selecting, evolving for captivity. Like there's natural selection for best captive traits.
Yep.
So really traits that are not suitable for captivity are getting purged by the keeper that goes, it's not going to breed, I have to get another one. And they eventually get another one, and eventually they breed, and so they're selecting for traits that make them do well in captivity. And I mean, people here will remember when black-headed pythons were hard to breed, now they're easy to breed. And even the one that everyone can't even believe that you could tell were hard to breed, and that was what we used to call Stimson's pythons. Oh, they're really hard to breed. And everyone these days go, what? It's stupidly easy to breed, you know, and so we were fiddling around 30 years ago or more with these pythons and trying to get them to breed. And of course, the ones that do breed are the ones that are going to breed in captivity. And the ones that don't breed are going to disappear. Those whatever traits they're carrying, same as mammals. Those first mammals, first wallabies that are caught out of the wild and released into a cage, the ones that run into the fence and die within the first minute of being released into their new cage, those scatterbrain traits are deselected, aren't they? And the ones that wander out of their carry box and go, oh, this is interesting, you know, are being selected for within minutes of being released into captivity, you know. But could you call keeper active, like Darwin's artificial selection, as a way of purging? Could you call that purging? Because say in the States of bearded dragons, there's thousands of generations of bearded dragons, and there's a few species. Apparently, we had a guest speaker years ago here, and he said there's some populations that are crashing, and that like the inbreeding pressure has finally reached its almost extinction point, you know. But I know new genetics does sneak out of Australia to go into the bearded dragon population, say, but if we're constantly deselecting these deleterious mutations, then we can sort of create our own purge. Is that a reasonable thing to say?
Probably. I imagine there's a little bit of purging going on in captivity of the deleterious stuff, and then there's adaptation to captivity, and you probably end up with a whole bunch of animals that do perfectly fine in captivity, but in the wild, and they probably won't.
We're not seeing wild populations of bearded dragons popping up.
Right.
We're like Sydney's even lost its original Barbados, pretty much. Like when I was a little kid around my area, Barbados were everywhere. You know, Southern Shire. Now they're extinct.
There's one more issue that seems to be happening, and I was going to ask you, and that is, you were talking about this sentiment that crept into the so-called conservation ideas, and that is pollution of gene pool causing outbreeding depression. But under that same potentially quite incorrect assumption, there's still resistance in potentially. Are you seeing that same sentiment with the movement of the giant desert skinks from one population to another? Are there people out there that go, oh, you can't do that, they're from different areas?
Well, I think people, because we're applying the tools to actually evaluate that, people are generally quite supportive of that technique. So we say, yes, the risk's low, or this is the best source population of translocating individuals from captivity. So we've got we've got data to point us in the right direction. But that view of risks of genetic pollution, I suppose, is quite entrenched across the board, and I think it's overstated. And I've seen it. I don't know whether it's changed, but I've seen it wielded as a sort of a rationale for not allowing people to keep reptiles because they might escape and cause problems. And I'm not convinced that really is an issue from a genetic perspective. Or a risk that's worthy of stopping people from keeping things that aren't in the area.
The other interesting thing that's happened is more and more people see captivity as the enemy of conservation. Better dead than in captivity. this because the big wildlife rescue organisations have put it into policy. If it can't be released, it's to be euthanized, and it actually mustn't go into even the zoo system now. So, for example, I think Territory Wildlife Park had 2 male red-capped robins or something, and there was one sitting in a wildlife rescue thing, and it just sat there, and it wasn't allowed to go to the captive population because it was, if it couldn't be released, it had to be euthanized, and that's what happened to it. It got put down. So that's the end of that species in captivity in Australia. But there's also even in New South Wales, in the zoos, the koala, they're not letting many, if at all, any wild koalas coming into the zoo system. So we are actually seeing inbreeding depression in koalas in Australia-wide now, pretty much. So, that many one testicle koalas are popping up in zoos right around Australia. One testicle koalas is a pretty sure sign of inbreeding depression, or inbreeding at least, yeah. It's coming.
Well, look, the only thing I can say to that is that my understanding of the zoo industry is that they're very well advanced when it comes to captive breeding, and by and large, as a generalisation, there are stud books, and they think very carefully about maximising genetic variation in their captive stock through, minimising variance in family size and managing through a stable sort of system. So from a genetic perspective, I think this industry is quite advanced in carrying out their captive management.
So you think that having those stud books, then the wildlife authorities and the various organisations would respect that and say, if you really need new genetics, then you clearly really need new genetics. But at this stage, we can't get through them, it seems.
I think it's sort of beyond the scope of this talk, but I think there might be an ideological gulf there that is part of the explanation, along with where one might agree with the science, and that's a whole different ball game beyond the scope of a single fellow like me.
So just with regards to the rapid adaptation to captivity, does that sort of make conservation programs like Toowoomba's corroboree program and Victoria's Eelis dragon program kind of useless? Like, can these animals be reintroduced?
They're very conscious. It's a very good question. It's a great question.
Yeah.
But they're very conscious of the whole adaptation to captivity issue.
Okay, so they manage that.
Yeah, we're onto it. But it's a great question. Glenn?
Yeah, just an extension of the sort of adaptation to captivity thing. It's one of the discussions that frequently gets brought up in the veterinary profession, and that reptiles should not be kept as pets because they are not domesticated.
Yes.
And it's impossible to domesticate something in just a few generations. It's taken cats all those generations to become domesticated.
Yes.
And there are no reptiles that have become domesticated because we've not been breeding them long enough. Therefore, we can't domesticate them. Therefore, they cannot be kept as pets.
Clearly makes sense to me.
Yeah. It's an argument pretty much from the veterinary profession.
But then you've got the cats that go feral.
Yes.
So is there
So it's a catch-22.
how many generations until it's domesticated? Is there any sense for that?
It depends on a whole bunch of things, like how many offspring they have. But what I can say is that there is scientific literature out there, peer-reviewed scientific literature that shows adaptation of captivity in just a few generations. You probably know more about it, right?
Yeah, don't ask me about it.
Okay. I have recently.
That would be really handy. That would be helpful for us as keepers to be able to.
Well,
It's hard to know because it depends on the strength of selection and there's a whole bunch of different things that come into it. How many offspring they have at the time.
Yeah.
But I think the take-home message is it can happen a lot more rapidly than people thought, and it is a real thing that happens. And yes, they are domesticated, if you like.
The Soviets did it with foxes in the 50s. They started selectively breeding for more passive traits, and then they saw the phenotypes be expressed with, you know, white colorations and blue eyes, and it's still today they have semi-domesticated foxes from these selected populations, and they saw, from what I remember, signs of quote-unquote domestication from 3 or 4 generations in. You know, now they're up to what, 30-something generations or whatever it is. But understanding that it happens, you know, we try to select for one trait, but inadvertently others will correlate with those, whether they're attached to the genotype or, you know, just happens to be whatever it is.
Tameness as well.
Yeah.
Foxes.
So I came to this talk because I'm interested in breeding a Gurnia. How would you like to see keepers manage their sort of populations? Like, how would, as a way to, you know, prevent inbreeding depression, how would you go about it as, you know, in a practical way?
You want to choose unrelated pairs. Actually, you know how I showed that slide, which was sort of along the lines of, well, is it familiarity? Because some of these species, they live in family groups, so the siblings hang out together, and do they go, hey, I grew up with you, I won't mate with you, or is it a genetic cue? I reckon the evidence is in favour of being a genetic cue.
Okay.
Based on, not my work, but some of the work that came out of South Australia, Bull's lab, and what we know from other organisms. So in that sense, the Gurnia's kind of almost got a built-in mechanism to help prevent that, because if there is a genetic cue, it's based on, often it's based on a part of their DNA that's important for immune response. So it's important for the offspring to be very different in this part of the genome, and it might just be that the Gurnia has an inbuilt mechanism that sort of safeguards against that. But anyway, we don't know that. So yeah, you just try to get as much information about provenance and pedigree. So it's I guess the point that I'd make is that it's really important, if you are a breeder, to keep good records and try to keep track of your pedigree.
I was going to say, it could be dependent. I don't know what the gene pool of some species are like in captivity. It might be unavoidable. That thought didn't go anywhere. Actually, don't worry.
It's actually a good thought. There are some taxonomic groups in captivity that probably have very few founding individuals, but you know, it is what it is. You do what you can with the information that you have, and I think if everyone keeps good information, keeps track of their pedigrees, and you use that information to maximise your outbreeding.
Remember, Adam, as part of Adam's study, he looked at my captive colony of Cunningham skins, which I collected when I was 13. Collected a few when I was 13, and the last female only died recently, so there's like 40 odd years in captivity, and I would have lived longer probably, but anyway, and you sample them all, and of all the animals, I remember there was at least half a dozen, 7 or 8 animals, and there was only one pair in there breeding. So yeah, one pair breeding. There was 7 or 8, something like that. Yeah, 7 or 8 animals, and there was just the one monogamous pair in there, and all the sons and daughters, and I think there might have been a brother or a sister or something of a brother of the female, the breeding female. And so there was all these animals that were related that were not having any mating success at all.
So
there's only one pair that was breeding in amongst them for year after year after year.
It's not even just like a preference thing. They'll actively refuse to mate with.
Refuse to mate.
Even if it means no offspring at all.
I'd say so. But don't forget, don't forget this. The female was always had her mate.
Yeah.
So who knows what would happen if that unrelated male was to go? I don't know. I'm not sure.
Awesome. Okay, put your hands together for Adam